Freshwater systems have been described as hotspots for biodiversity and endemism, with freshwater fauna experiencing high rates of decline in the 20th century. Freshwater is thought of as a very important and vital resource to many organisms, including ourselves, humans. And the growing demand on the freshwater that we have here in Central Texas, with the increasing human population, poses quite a bit of threat to many endemic species that occupy this aquatic environment. The Edwards Aquifer is an important source of freshwater to many residents and is directly or indirectly supplies water to major cities including San Antonio, San Marcos, and even Austin. The Edwards Aquifer supports a number of aquatic habitats that harbor multiple endemic species. Some of these species inhabit the inaccessible regions of the aquifer, and therefore they have relatively low detection rates and have been proven problematic to sample and just to kind of fully understand the abundance of these organisms and their distribution. So several endangered and threatened species inhabit these subterranean regions of the Edwards Aquifer, mainly the neotenic salamanders here in Central Texas. We have only been able to sample them sporadically, either through spring outflows or wells. However, the relatively low detection rate of these salamanders, combined with the inaccessibility of their habitat, makes sampling very difficult and warrant perhaps a new approach to investigating their distribution. And so what this project aims to do is to collect DNA of these salamanders from their environment. It has shown to be sensitive and an efficient way of detecting aquatic vertebrate DNA. It's been used in rivers and canals and fast-flowing streams, but there's very limited amount of testing or use of eDNA in karst aquifer environments. So our previous work on the Edwards Aquifer has shown that eDNA methods can be used to detect presence of groundwater salamanders of the genus Eurecia, found here in Central Texas, in the Edwards Trinity Aquifer system. Subterranean organisms are among the most cryptic and least understood of all Texas species. The species that inhabit caves and aquifers are more difficult to detect and collect than their surface counterparts. Particular subterranean groups of conservation concern include the salamander genus Eurecia, for which there are a number of endangered species like Eurecia rathbuni and Eurecia waterluensis, which contains at least the entire group of the Texas Eurecia, contains about 14 to 15 species unique to Texas. They're all listed as species of greatest conservation need in the Texas Conservation and Action Plan. 2 federally listed as endangered, 4 as threatened, and 5 are under review. Critical habitat has been established for 3 species but remains to be finalized for others. Due to the paucity of information on distribution and habitat requirements, it is uncertain how to determine the best plan of action for conservation for some of these species.
Hi, I'm Tom Marshall. I'm a PhD candidate in David Hillis's lab at the University of Texas at Austin in the Department of Integrative Biology. To filter our eDNA samples, we use special filter cartridges that are designed specifically to filter eDNA. The filter paper is inside this cartridge, and there's also a desiccation chamber inside the cartridge that allows us to store the filter for several weeks, even up to several months, without any kind of refrigeration until we're ready to extract DNA from the filter. So to start the filtering process, we simply remove the cartridge from the packaging. We connect one end of the cartridge to a hose that's attached to a pump. This is to pump the water from the sampling bottle. We then connect the other end of the cartridge to the water bottle, turn the water bottle over, start the machine, and then water will begin pumping through the filter cartridge, now through the hose into the sink. So this way the water is draining through into the sink, and any DNA that's in the water is attaching to the filter inside the cartridge. Once the filtering is done and all the water is filtered through the hose, we turn off the pump, disconnect the hose from the filter cartridge, throw away the water bottle, and put the cartridge back into its packaging, where we can keep it at room temperature until it's ready for DNA extraction.
Hi, my name is Ruben Tovar. I'm a PhD candidate in the Department of Integrative Biology and in David Hillis's lab at the University of Texas at Austin. So we start by having our samples on the ice. We have our assays, which include the forward primer, reverse primer, and probe. We also have the TACMan master mix as well. So before we start, we need to make a master mix associated with the assays that is specific for our species. So to do this, we are going to make our master mix by adding these quantities together that I have already calculated based on the number of samples that we have. So for the TACMan master mix, I will need 52.5 microliters. Set my pipette to 52.5. And I will pipette 52.5 of my TACMan assay into my master mix. I will then pipette 11.69 microliters of water, DNase free, into my master mix. We'll then add the forward and reverse primers, 0.42 microliters of each. Here is the forward into our master mix. And our reverse into our master mix. Finally, we'll add the probe. The probe is 0.21 microliters into our master mix. Here's our probe. Into our master mix. Then take the master mix and vortex it for just a couple of seconds. And now we are ready to aliquot our master mix into our plate. I have the qPCR plate here in my hands. I'm going to label it the species of target. One species is Eurecia neotnes, and the other is Eurecia latitans. I've abbreviated the names here. I'll also label the amount associated with these assays. Since we are running a serial dilution, we know how much is in each of these DNA samples. So I will write them here. And finally, the last row will be dedicated to a negative control. After I've done that, I am now ready to add my master mix into the wells. There's going to be 9.32 microliters of master mix added into each well, and 5.68 of the DNA extract for a final volume of 15 microliters. So first, I'll add the master mix, 9.32 microliters. Being very careful not to spill into other wells. I am now ready to add my DNA product. We are going to add 5.68 microliters of each of our DNA product into the wells. So here's my DNA. So I'm pipetting up and down a couple of times to mix the DNA product with the assay. There we are. Now I'm ready to place the covering film on top of our qPCR plate. It can be a little bit finicky. Trying my best not to touch the inside of the film. I'll gently set this over the plate, smooth it in the middle, and kind of smooth it out towards the ends. Usually take a pen, just kind of rub over. Make sure it adheres to the plate. Tear off the perforated edges. Okay, now we are ready to place our plate into the qPCR.